Thank you. Good morning to everybody. Yeah. Now, let me make my presentation basically on some of the non-chemical aspects with respect to the tea shuttle borer, what we have experiences in Sri Lanka. First of all, welcome to the Tea Research Institute of Sri Lanka, a wonderful working environment for shuttle borer, being this particular pest has been in existence for the last several decades. And during this brainstorming session, I'll just take you through two aspects. One is the invasiveness of the pest, and secondly, some of the experiences what we have on non-chemical control. Now, ladies and gentlemen, when you talk of invasive animals for tea, we don't consider shuttle borer as you consider today as an invasive pest, because we have been having this problem for ages, and we are on the process of making several attempts. But on top of it, we have some other invasive animals in the tea sector, starting from various other animals in the tea sector. Why I am so concerned about harping on the non-chemical methods in tea, or making the tea shuttle borer control. Tea is considered as the main economic crop for the country, and more than 95% of the tea is being exported, and we have restricted usage of pesticides while harnessing more of non-chemical methods. So that is the main reason why we are so concerned about the pesticide usage, and tea shuttle borer is one such pest affecting tea, while we have many other pests from top of the foliage down to the root. Okay, now this looks like a real tea plantation. You can see. It's a fully covered land with tea, and when you go closer, you will see a nice looking foliage full of branches. Per bush, you may get about 60 to 80 developed branches, and per acre, you will have about 5,000 plants, right? 5,000 bushes per plant, right? I mean, for you to think how difficult if this problem is going to be cured, if you are harping on one single bush in terms of that way. Right, now on a bush you get different sizes of branches, and you will see young as well as mature branches, and shuttle borer, this particular species, will prefer branches of this size, right? Branches of this size, where we say for our convenience we say pencil thickness size branch. So even for assessment of damage, forecasting the damage, we consider these sizes of the branches for our assessment purposes. The damage goes on from young stage up to the mature stages, but not on dry wood, right? Not on dry wood, mainly on the living plant. You can see the damage extended in different life stages of the stem. The first attempt what we do is, as I said, the borer population is found in the environment or in the ecosystem. All what we are targeting is to look for cultivars, resistant tolerant cultivars as the first step, And secondly, prophylactic stem protections, where we can stop the beetle going into the stem, right? I mean, that is the first step. And then secondly, when we realize the bush infestation, certainly we do surgery pruning, or take off all the damaged portions, and that is another aspect what we advocate. And thirdly, making the plant healthy and improve the plant's vigor through various good agriculture practices. I recall some of the discussions yesterday, expecting the beetle would do a fairly big damage when the plant is under stress, right? So this is one such approach what we practice. So we advocate the growers to go for the good agriculture practices, starting from nursery going up to the mature stage. I mean, cover all aspects like fertilization, taking the harvest properly, and control of other insects and pests or other pest damages below threshold levels. The sanitation pruning I was talking, the tea, once in 3 to 4 years time, you take off the branches or mature branches out, and then our main attempt is to make the plant form the plant to a bush, right? So that way we'll take off the branches out, and then, as I said, a surgical operation is done to take off all the affected portions. And in the meantime, take off all the branches what we have taken off, or prune branches, and then chip them and bury them. We expect two objectives. We have two objectives here. One is to take off the inoculum, right? The pest inoculum 0, and secondly, when you incorporate all the chips into the soil, we expect the organic matter level will go up, right? So that's another way of making the plant healthy and vigor. And of course, following pruning or taking off all the affected portions, we do a wound dressing, a simple non-chemical wound dressing. That's a spray lime, right? Just a hydrated lime spray onto the bush, right? And we consider this as, again, when you spray lime, we are experiencing a global change. Climate change now, and this acts as another climate change mitigation, where you don't get splits of the branches, right, or cuts branches. That's another objective what we have, and of course we are developing certain other stickers onto this, so that that's going to be a two-way action, while reducing the secondary infections onto the cut surfaces, as well as mitigating the heat stress onto the plant. And after about say few months time, say 8 to 10 months time, you will get the branches booming up, and that would be the stage where the borer populations would attract to the plant. So that is where we make the spraying, right, just to protect the stem from the beetle attack. Right, now little bit of biochemical and physiological basis in identifying the susceptibility or tolerance of the tea cultivars. You know why you are drinking tea, mainly because of some of the polyphenol compounds, right? That is what you get. That's what you expect from tea. So in the leaf you get the same chemicals. So it's the case with stem and the other parts of the plant. The basis, main biochemical basis behind the susceptibility or tolerance against the pest in tea is related to saponins and to a great extent, and also for polyphenol contents in the plant. And there are studies to say that relative amounts of spinasterol and saponin have been related to the pest tolerance, right? And at the same time, this has been identified, or this has been proven to say that some of the substances are inferred with the hormonal activity of the beetle during pupation, right? So I mean that's something what we know. And we have different, we got about, I would say, more than 30 different varieties, cultivars, with high degree of tolerance. And certainly those plants or those varieties will pose in high levels of these chemicals, right? So that's something what we know, and we are in developing a new variety. We are looking for such biochemical and physiological basis. I'll come to such chemicals later. And some of the another important area where we need to understand is, in between the nodes, right? In between the nodes, normally beetle attack is more prominent towards the leaf, right? Towards the leaf, where you get more of such chemicals, right? More of such chemicals when the borer recognizes the area, so that they come and attack the susceptible portion, right? But in between the internode, you get less of those chemicals, and then you get less damage there. So that's something we know of. And in addition to saponins and spinasterol, we get some understanding on caffeine. Caffeine is low, of course, in tea, right? I mean, mainly you get polyphenols, but polyphenolic compounds, phenolic compounds. But when you have caffeine, then there again there's a degree of susceptibility. And of course, some of the most important volatile compound, right? I mean, tea again, as I said, tea is marketed, or tea is recognized for flavor, right? For flavor, for flavonol compounds, certain volatile compounds are responsible, and there are 5 important compounds identified, and these have been recognized as attractants for the beetle, right? So indirectly, if you are going for a high quality tea variety, certainly the borer damage would be very much high. So it's some kind of a pricing for us to decide whether we are going for a high quality variety or as a balance, fairly high quality and fairly a good cultivar with respect to high degree of the tolerance. I was talking about this, and infectious stems show increased levels of caffeine again as a defense mechanism, right? In the stem, the levels of caffeine will go up as a defense mechanism. I think we saw some of the examples yesterday here on avocado. Maybe some other chemicals would go up. Maybe in tea, only caffeine and polyphenol we have seen after infection the levels will go up. Ladies and gentlemen, now let me give you some success stories with under organic tea cultivation what we have experienced. Organic tea has been introduced by Sri Lanka. I mean, Sri Lanka pioneered in producing organic tea, and we have long experience on maintaining pests and diseases below threshold levels using non-chemical methods, of course, without any synthetic inputs. So we have a long-term trial TRI, Tea Research Institute Organic and Conventional Comparative Trial, where we have observed with respect to shuttle bowl I'm talking now. Under organic systems, we have studied the soluble sugar levels in the stem, and we found that the sugar levels are comparatively low compared to the conventional treatment, right? Conventional treatment where you advocate synthetic methods, right? Synthetic methods or synthetic inputs, fertilizer as well as pesticides. The reason what we noted, the physiological basis what we noted behind this lower sugar level is the stress, right? It's a stress due to imbalanced fertilization in the plant, right? So more water is found or translocated in the conventional system, while under organic systems the water penetration or water translocation is very much low compared to the conventional system, so that the compounds in the stem will be fairly low. And we did a study with the sugar levels and the same extracts, and we introduced the ambrosia fungus, and we noted that the under organic system the ambrosia fungal growth is inhibited or low compared to the conventional system. So as a result, even the limited number of beetle would do a fairly big damage under conventional system, while the organic system would do little damage. And with respect to the important volatile chemicals I was referring a little while ago, you can see in the conventional systems, you can see the level, right? The level or the peak area, I'm just giving the peak area or the levels of these such chemicals, fairly high compared to all these three organic treatments, right? So simply under conventional system, the attraction to bora is high compared to the organic system, right? Organic system where the volatile chemical concentration is fairly comparatively low. So is the case when we did a bioassay with the stems taken from organic as well as from the conventional system, the beetle attraction was fairly comparatively low compared to the conventional system, right? Now the beetle were so keen to go for conventional stems compared to the organic stems. And ultimately, I mean, what is important is how does the damage occur on both organic and conventional stems. You can see, I mean, before I get into the data, we did again, we pumped a dye on the stem, and we noted in the conventional system the dye went there were several blocks. Sorry, the other way around. This is the conventional. This is the organic. In the conventional system, because of the bora damage, the dye did not move fast, and in the organic system, the dye went without any hindrance. Right, the damage levels are very much low under conventional system. And you can see the total damage when you compare with the conventional system, the resultant damage under organic tea and under organic system was fairly low, right? So I mean that was the evidence we have in organic system. And in the meantime, we studied the histological studies or the stem structure in stems. Of course, I mean we studied. I mean we have done this for leaf pest and root pest as well. But this is with respect to shuttle borer. We noted under organic systems the tissues, the xylem tissue thickness is fairly high compared to the conventional system. That will act another hindrance in making the borer damage lower, right? So I mean that's one example what we have seen. We have seen the same, similar scenarios on leafy pesticide, leaf diseases as well as the root diseases, right? So that's about the histology. To brief you what we have shown or what we have experienced with respect to non-chemical method. Certainly our main approach in tea shuttle borer management is to look at the critical points where the borer would get into the plant, and then we encourage the good agriculture practices so that the borer damage would be minimized, right? The borer invasion would be minimized rather than waiting for the infected bush and infected plant, so that for us to go for control method. In the meantime, with your understanding here in US, we studied we went around some of the close by areas where again we saw the pest damage occurring on avocado in Sri Lanka at present, unfortunately, and so is the case on durian, right, and rambutan, right. So I mean those are some of the. I mean of course we knew these plants as hosts, right. I mean we have about 140 odd different plant species as hosts, right, in addition to tea, but we don't consider. I mean we have not seen the beetle completing the life cycle or doing a big damage, but now we have added added the host range with these three important plants or important trees to Sri Lanka. And with my presentation, with my experience, right, what I presented now, I think we would help you in studying some of the host range clearance with I mean we were trying to clear the beetle as well as the fungus anyway, but at the same time we should look at some of the host range clearance. Certainly that would give us, the whole team, a good exposure. And of course resistance and tolerance mechanisms in your plants, in your species what you are so concerned about, and also to recognize some of the critical points in avocado cultivations here, right. I mean as we have experience in Sri Lanka, what critical points what you can recognize so that we can advocate or we can target some of the control practices. And of course good agriculture practices for better control, in addition to pesticides or pesticide injections or whatever. And I could not tell you some experience what we have with high potash, right, using high increased potassium in fertilization or reduced levels of nitrogen, right, what we advocate under organic systems, right. So that's another area where we need to work on. And collaborative links and exchange of expertise, as what we did or we started now, will compare to the previous experiences. And thanks for you for giving us an opportunity, for Sri Lanka, an opportunity to relook at the biology, right, and relook at the behavioral aspects of the tea shuttle borer. And because I mean we have been not forgetting, but we have been neglecting that area for some time, because we don't see much new pest scenario. But certainly with your initiation now, certainly we'll get together and look at some more on that. So with that, ladies and gentlemen, thank you for your attention. Hope I gave you a few comments as an insight to the problem. Thank you.
I'm curious about the fact that the beetle attacks such thin branches on the tea plant in comparison to other tree trunks and, wider woody branches. Is this an unusual aspect of the biology of the tea shot-hole borer in Sri Lanka, and is it specific to, say, tea? Or do you see this in other crops where you grow, say, a shrub or a bush? Does the beetle also attack the thin branches?
Yeah, of course, on tea, that is the susceptible stage, right? That's the susceptible stage. As I said, the biochemical basis in different sizes of the branch has been well understood, right? And of course, the attractants as well as other compounds, right? But to give you some other examples within tea, we know. I mean, we have seen damages on other sizes of the branch or sizes of the stems, but this is the most susceptible stage. But when you go to avocado, rambutan, or any other crop, certainly we get additional sizes or additional stem sizes. Yes.
In tea, do you find that the fungus grows down from? Does the fungus grow at all? Does it spread in the tea plant after? From
no.
where it's just in the galleries.
No, in the gallery itself. Yes.
Okay.
But recently we encountered another symptomological difference. Just similar to some of your experiences here on the stem. Just an oozing out of some liquid. I mean, dark color liquid. That's all, right? I mean, we don't expect anything to come out from the gallery, but just a symptomological difference. Yes.
Okay. Then one other plant. You had a question. You had durian was another host, and it does reproduce on there.
Yes.
And what was the second one?
Rambutan.
Okay.
I mean, just close to lychee. Yes.
Okay, one more quick question, and we move on.
So, do you do a lot of pruning on the tea plant? So, do you see any attack on the pruning wounds by the beetle?
No. But through pruning, we don't get any attack. We don't attract beetles, so we don't get any attack to the pruned cut. Yes.
Ruben, last word.
Has anybody tried injecting or introducing saponin into the tree as part of the organic farming, not only depending on the natural occurrence of the saponin in the tree?
You mean injecting insecticide, right?
Just saponin.
Oh, right. Okay. No, I mean artificially spraying saponin.
Yeah.
No.
Spray or inject systemically, no?
Sampath, do you have any experience? No. But
now, tea is, we consider tea as a health beverage, and we are not supposed to inject anything and to modify the constituent. So we consider it is a natural beverage, so we have to go as it is, right? So saponins or injecting of anything or genetic modification is not possible with tea, right?
yeah, just one comment. I mean, I'll share some of the experience later. But we, as Sampath said, I mean, we do not do anything on the field, right? I mean, spraying anything, but we do study certain incorporation of such chemicals on artificial diet, right? Where we have experiences, as I said, the same phenomenon what we experience in the field, we reproduce the data similarly on artificial diet. I mean with increased saponins, increased polyphenols, increased levels of pesticides, right? That we have observed.
Okay, we should move on. Thank you.
Thank you.